This is one of the questions that comes up quite frequently is customers and clients of ours have questions about how do they analyze cell rolling and cell rolling events. And sometimes before that, they'd like to know why that sort of thing is important. So the agenda for today, and hopefully we'll be done in about 35, 40 minutes, is a brief introduction to BioFlux. And then we'll talk about cell adhesion and cell rolling, just as an introduction on a broad level. Talk about previous publications, and then go through a cell rolling exercise. And that will finish us up for the day. So a little background about Fluxion. It was founded in 2005, headquartered in South San Francisco, California. And we've got worldwide distribution. Actually, now we have distribution into Australia. And the BioFlux has one unit in South America. We also have units in the Middle East, including Qatar and Pakistan. Early on, we won 5 NIH grants to develop the technology, and were awarded the R&D 100 award 3 times for the respective products. The BioFlux, which I'm talking about today, has to do with live cell imaging under physiological conditions. The IonFlux is an automated patch clamping system that allows for an automated high-throughput electrophysiology readings and really excels at ligand-gated channels. And the IsoFlux is a next-generation device for cell isolation, for isolating rare cells such as circulating tumor cells. The importance of shear stress and perfusion has been known for some time, most notably in the biofilm community and the platelet community. That in the blood vessels, blood perfuses through at a certain rate. In small veins, that's low, about 1/10 of a dyne, and in the aorta, it is as high as 20 dynes. And in pathological areas, it's about 200 dynes or more, in that ballpark. The reason this is significant is that when you study the behavior of both blood and the interaction of blood onto endothelial cells or cells that would be in the blood, it's important to be in these physiological ranges. This is an example of whole blood that's been labeled with calcyan-AM. So platelets and white blood cells are labeled. Under low shear conditions, there's really just a basal amount of adhesion onto the surface of the plate. All things being equal, everything being the same, under high shear conditions, the blood coagulates completely. And the reason this is significant is that this is one variable that basically initiates the clotting cascade. This is an example of tissue culture where the top panel here are cells grown under static condition. These are Hubeck cells. And you see basically what you would typically see in a tissue culture dish when you plate down cells. But they don't look physiological. The panel below that shows the same cells that are perfused at 2 dynes for 16 hours. And you see a physiological rearrangement of the cytoskeleton, which you see on the right with the staining for actin. Another significant, an interesting point about this is that it's not just what you see under the microscope, but there's also molecular changes that occur, changes in transcription and translation and post-translational events that you would miss if you didn't culture your cells under a physiological properties. Static well plates are really great for inexpensive cell-based assays, but they have a significant limitation in their ability to perfuse media, and you can do basic cell adhesion experiments, but they're not physiological, and you certainly can't do cell rolling experiments. Conventional flow cells have been used for cell rolling experiments for some time and for thrombosis experiments, The biggest limitation here are the amount of media that you need, the amount of blood that you would need for this type of experiment, and the control that you would have over the flow with this type of pump. And a perfusion system like this would not give you a precise control over the shear stresses that you're putting on your samples. The BioFlux combines the throughput of a 96-well plate with the high-quality data of a flow cell. And so essentially it's an array of flow cells that are put together. Several advantages include precise control over shear stress, the ability to use significantly lower amounts of liquid too. You know, one example of that is if you're doing a mouse thrombosis assay, typically you need 300 microliters with a BioFlux assay plate, whereas the same type of thing with another system, you would need 3 milliliters. So instead of using 3 mice for a traditional assay, if you're, depending on how big the mice are, you could actually get 3 assay points per animal, which will increase your efficiency by 9 times. The BioFlux 200 includes the pressure controller that has pneumatic electronic control on it, the microfluidic plates, and the BioFlux software, which controls the flow and also has some basic analysis functions. The BioFlux 1000 system adds on an automated Zeiss microscope that has an automated stage, automated fluorescence, and our software, which is based off of Metamorph, which takes care of high-level acquisition and then high-level analysis. In comparison of the approaches that we like to think of, the throughput as being quite high with a static well plate, and the physiological relevance quite high with flow cells, and the BioFlux is somewhere in between. The principle for how the BioFlux works is that you've got a microfluid plate, which is on a SBS-style format, where one well is basically serves as the inlet, and pneumatic pressure from an interface comes down and pushes the liquid from one side to the other, pushing media, pushing things like coating proteins or cells that would roll along the surface from one side to the other under controlled conditions. And you would use an inverted microscope, and the bottom of this plate is a number 1.5 cover slip, which is 170 micrometers. The characteristics of the way the plates run is that you've got an inlet, a serpentine, which is essentially a pressure resistor, and an outlet. So if you think of a massive a beaker of media here and a beaker of media on this side, that would be a way to visualize this. In this case, you've got 2 channels that you can image at the same time. This is an example of the flow velocity profile that goes along with the system, and you see that in the viewing channel that the flow is very even and controlled. The basic operating principle is that you load the microfluidic plate, you attach the interface, and then you run the sample. The cell rolling has actually become quite popular in the past few years. Back in the 1800s, Eli Metchnikoff, which he got the Nobel in, I think, 1908 for cell-mediated immunity, said, next to leukocytes, the vessels and their endothelial lining play the most important role in inflammation. And what he meant was, aside from looking at the individual cells themselves, looking at the interaction between the endothelium and the cells in circulation was a significant thing. And we're seeing a lot more in terms of phenotypic assays that come out, instead of just looking at cells and noticing whether or not they express a particular cytokine or message or protein. Another facet of this is circulating tumor cell analysis. That the classic definition of a CTC is EpCAM positive, cytokeratin positive, nuclear DNA positive, and CD45 negative. And that's the kind of protocol you would take towards isolating the cells. But researchers also want to see how aggressive these cells are. And what would define a circulating tumor cell as being active is being able to adhere to the endothelium and then migrate through and cause a secondary tumor. And the third is stem cell adhesion. I'll talk some more about this later. But stem cells, if you're going to get a stem cell therapy, the cells need to be able to home, attach, and then transmigrate through in order to do the respective function. And as a result of that, if they're not adhering at the right rate or the right time, they're just going to be lost. They're either going to just die in circulation or they'll be eaten by the lymphocytes. This is an example of stem cell homing and the desire to study this type of event. This paper came out of Jeff Karp's lab out of MIT and Harvard. This particular figure shows the velocity of cells rolling versus the shear stress. So they did a quarter dyne, half dyne, 1 dyne, 2, 5, and 10. And what they were comparing were HL60 cells, which are a classic cell rolling example, to mesenchymal stem cells. That they wanted to adhere and roll sort of at the same rate. And so they transfected and overexpressed different proteins, including this SLEX and PSGL1. And you see that didn't do much of effect compared to the native MSCs. But when they double overexpressed both of these transcripts, they showed that the cells behaved more like HL60s and would be better for stem cell transplantation. This is another figure out of that paper where they were looking for a more normalized and optimal range of the velocity of these cells. And in native cells, they sort of had this even distribution. And in the double transfected, he showed that they were able to tighten the range of the way that the cells rolled and the way the cells moved. This is a video out of their lab which shows HUVEC cells that were plated in a monolayer and then HL60s that are rolling along. And you can see the cells in different areas. These cells were a little bit clumpy when this image was taken, but you can track the individual cells as they move. This is another video taken at the University of Manchester showing a HUVEC monolayer on the surface and then HL60s rolling over top of it. These are cells that were treated with TNF-alpha 4 hours before the experiment. TNF-alpha typically serves as a positive control for activating these the adhesion proteins in the endothelium, probably through a post-translational event, because things happen within 4 hours and it's transient, such as HIF, for example, or AKT, though AKT happens a lot faster than 4 hours. This is an example of the exact same cells, but were run with, as a control. These cells did not have TNF-alpha treatment, and as such, they show minimal, if no, binding at all to the same monolayer. And actually, if you see here, the flow is actually paused for a moment so that I could show the researcher, the graduate student in the laboratory, that I didn't put a video on his computer, that actually this was a live experiment taking place. So we paused it for just a moment so you could see the cells, and then started the flow back up again. This is an example of HL60s rolling over E-selectin. And so another option for looking at cell adhesion, instead of cell-on-cell adhesion, this is cell and ligand adhesion. And you can coat the surface of the BioFlux plate with any type of protein that you'd like to, as long as it's in solution, and be able to study the protein specifically instead of the protein as in the context of several others that you would find with cells. One of our customers that we recently installed a system to had published a paper before they purchased this, and we basically repeated their findings. And this is a summary of what took place. We took high shear plates, and these are plates that go between 0 to 200 dynes. They were coated with the A2 domain of BWF. This was a mammalian recombinant protein. And so the plates were coated, they were rinsed with PBS, and then whole citrated blood was perfused over the channel and incubated for about a minute. And the blood was rinsed back off, and really only the platelets remained. After that, PBS was perfused over top of the platelets at 60 dynes, and this was imaged at 40x in bright field. And so I'm going to switch to, this is our software we call Montage, which is based off of Metamorph. And this is the video that was taken at the Houston VA. And you can see that there's a variety of morphologies to these platelets, that some of them are quite flat and others have a more flattened out, or more tubular type structure. And this goes on for, so this is taken for 40 seconds, 500 frames. It's a pretty big video. So one of the strategies to not overwhelming the system is to break this into a few parts. And by doing that, we can analyze subsections of the data. So I'm going to take a section here. This is about a fifth of it. And I'm going to go Edit, Duplicate, Stack. And that's going to give me a section here to work with. I can close this out. So still 500 frames going from left to right. And since the cells really don't go up and down, but just go left and right, there are very few events that are missed by doing this. Then I'm going to go to Standard, Process, Morphology Filters. And I'm going to start by inverting the image. This was taken in bright field, and I'm going to invert the image so it looks more like a phase contrast image. I've selected the source file here, and I've selected all planes, and just hit Apply. By the way, if you have any questions as we go along, feel free to write them in the question box. There's a couple analysis steps that take a few, about 2 minutes with the computer, and it's a great time to look over what you've got. So here we have the cells that have been inverted, and then I'm going to take and further modify this image by filling in the holes. And that just cleans up this so that the software can recognize the cells as they move. So you can see in comparison between the three planes, this is what we started with. This was the inversion, and this is with the holes filled. Nothing, data hasn't been changed, it's just we've made it a little bit easier for the software to be able to recognize the cells. So I'm going to close out these two. I'm going to go with this guy. I'm going to go to Apps, Multidimensional Motion Analysis. This is, most people when they get the BioFlux 1000 purchase this item. It's actually not that expensive to add on if you already have a program in place. And I'm going to do, so this is what they call the MDMA. My source image is going to be holes, which is this image here. And I'm going to do an adaptive threshold. And this basically stipulates, size of the object looking at, has a relationship to the intensity of our background, and talks about the travel distance between frames, meaning if the cell is moving from one side to the other, that we allow a give of 50 pixels, which can later be translated to micrometers very easily with calibration. I'm going to preview what the cells will look like, and you see most events are all captured here, including the long cells, or the round, the more tubular-like cells, and the round ones. Press OK, and then hit Start Tracking. And this will take just a few moments to do. And while we're waiting, I'll put back up the protocol that we used to look at this initially. It's almost halfway done. If you can't think of any questions now, feel free to email me either at support@fluxionbio.com or brian.haynes@fluxionbio.com, and I'll be happy to get back with you. Okay, we just finished up here. This is a the tracking that you see, this sort of colorful montage. And we're going to go to display data, which is in the lower left-hand side here. And we've got a variety of options to look at. So each object is annotated here. And we can start by hiding all tracks. And that allows us to see the cells as they appear. And then, so what we're seeing here are the XY coordinates for each object, and as they appear on the screen. So in some objects, for example, have a span between several hundred frames, or at least 100 frames, which show that they basically traveled from one end of the plate all the way to the other. And in this case, we can see this is travel at the top here. This second object actually had quite a bit of travel. It started here and moved all the way over here. In addition to position, most people will look at two specific things, either distance, which is limited to the length of how far you're imaging based off of your camera and the objective used. And the second one that they'll use is velocity. So with the distance, you would simply add up the distance and have that in a table format. For velocity, you would average these velocities. And I have a question here. Let me take a look at this. So the question was, what is the shear stress that the cells are exposed to before they get into the viewing channel? And so the viewing channel is 350 micrometers wide, and the serpentine, or the area outside of that, is 90 micrometers. So it's a multiple of 3.8 times whatever you're typically setting it to. So for example, if you set 1 dyne for your flow, that in the serpentine, the cells would be exposed to 3.8 dynes for that time period until they got to the one end. Now that's with the low shear plate. With the high shear plate, and these were done with a high shear plate that goes between 0 to 200 dynes, that's not an issue because the cells start out actually in a wider area and then flow down into the active area of the viewing channel. So they go, we haven't calculated this because it's a little bit of a conical shape, but several fold less in terms of shear force until it gets to the viewing channel, and then it goes to what's set with the program. So getting back to the data here, you can look at these different objects, and of course, as you see them, if you really want to, you can go through and decide if an object is a visual abnormality with the software, or, for example, here where you don't see anything, did it just not move? And that's very possible that you see the object and then either it flew off between frames or it just stayed firm, and then the object wouldn't move in that case. It would have adhered, but it wouldn't move. As you move down in the objects, you'll see that the activity takes place later on in the frames, such as this one. This one is here, and it takes place pushing frame 95 and forward. Next thing I'm going to show you is something called a kymograph that is more popular in the old school technologies, but gives you a visual representation of cells and as they move. Just briefly, a kymograph is referred to as a wave writer, which is a device that gives a graphical representation of a spatial pattern over time in which the spatial axis represents time. If that's not a tongue twister. So this is an example of a pizza being built. And as the process continues, it starts, this is basically time point 0 at the top, and at the very bottom is the end point. So as you start out, you've got all dough, and then you've got the pizza sauce, then you've got the cheese, and then you have the various toppings, and you see that represented. Anything that really sticks out winds up getting pulled out in this graph. So we're going to do the same thing here. I'm going to take a single line and trace it from one end to the other. And then go to MM Standard, Stack, Kymograph. Now this is set for a line width of 15, and if you want, you can basically get more real estate or less real estate. This is about the width of what one of these platelets would be. And my source image is holes, and I just press Create, and it will build. So what this shows here are, as a function of time, at the top is time point 0, and this correlates to the cells that would roll. And as you line it up, you see from time point 0, for example, this cell rolls along and then falls away. There's looks like an artifact here. As we run this, no, it's not an artifact. This is a cell that's adhered and hasn't moved. You see it starts at the very top, and then as you continue the whole space, it doesn't move, and that's represented here by this straight line. The rest of these show really the velocity, and if you wanted to, you could take the slope of this and then have a little bit of a quantification. This line can be moved and then reanalyzed. So this should be similar but not the same, because it was in a different area, but the cells were all from the same grouping. Now, in theory, if you compared this to another control group or to another experimental group, these kymographs should look different. Excuse me. Last thing we're going to do in terms of analysis is look at the shape of the cells to give a representation of the type of population that we have. In order to do that, we need to threshold the image for light objects, because we have light objects here. And you can see that the threshold can be modified a little bit, but the idea, of course, is quite simply noise over background, or signal versus noise in this case. And you see we've got the different objects here. We go to integrated morphology analysis, and we're going to do a couple things. First of all, under our measurements, we're going to look at total area, shape factor, length, and breadth. And we're going to filter out the cells in terms of total area that we don't want any background, and we so in terms of these pixelated images, and if for some reason there's an artifact, we don't want anything over 3,000 in terms of total area. But we're really interested in looking at shape factor. Under preferences, we're going to fill in the holes in the objects so that an object like this will be filled in because we know it's one cell. We hit measure, and we see all the objects that turn green are the ones that measured. This background here is not measured. And then different things you can look at. This is looking at the shape factor. So as the cell becomes completely round, the shape factor is 1. As a cell is completely as a rod, like for example for bacteria, the shape factor approaches 0. And so in a perfect situation, which is pretty rare in biology, perfectly round cells would be a 1, and perfectly rods would be a 0. And you see a distribution of the cells in this case. You can also look at length of the cells, breadth of the cells, and total area of the cells. And you see there's a fairly even distribution between these, but there's some patterns too. And when you go to present the data and you put this into a manuscript, these are things that would be helpful in showing at least a normalization to what you're doing. That sums up our discussion on an analysis workflow. So we talked about the chymograph. This is a partial list of our customers. Some of these were early adapters, such as Stanford, and others are more recent additions. And easiest way to contact us is either support@fluxionbio.com. Fluxionbio.com has our literature and our videos, and this webinar will be posted eventually for future reference. I'm happy to take any questions at this point, and otherwise we'll wish you either a good night or good morning, depending if you're in the US or in Asia.
Thank you, Brian. If you have any questions, you can write them down in the question box, and Brian will be happy to answer them. We'll wait for a few minutes for the questions, and otherwise we're just gonna close up, and we'll take questions by emails later on. Okay then, Brian, thank you very much. Thanks everybody for attending, and we'll hope to see you soon in a new webinar. Thank you very much. Bye-bye.